Hello, welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 19, where we will talk about the RNAi-mediated replacement of morphine with non-narcotic alkaloid reticuline in opium poppy. So, this is a very interesting, this is indeed very interesting job what scientists did. And one important thing I would like to tell you that this work again was done by the Australian group, that is a collaboration between CSIRO Division of Plant Industry and the Tasmanian alkaloids. So, let me go to the board. So, the concept is one concept only we will discuss here in this class. This will be on this how metabolic pathway engineering can be done using the RNA interference technology. So, what important here is that this paper is published in a famous journal which is called Nature Biotechnology. So, Nature Biotechnology is very famous and they publish those papers which describes or which report for the first time something surprising fact and or some outstanding observations. So, this paper published in the year 2004, although now this is more than almost 10 years old, but still this is very relevant. So, that is why I have chosen to discuss this paper. And Robert S. Allen is the first author, and in this group Philip Larkin is also there, and this is from the Australia. Okay. So, what they have done is, what is the case study? So, this maybe we can call this as case study 5. RNAi mediated metabolic engineering. I just give this name. So, here they have targeted coordinone reductase COR, coordinone reductase. So, what I have told, COR is basically the enzyme coordinone reductase which converts coordinone to coordin to coordin and also morphinone to morphine. And what I have said that this requires NADPH for its function. Okay. The variety they have used, which was basically the Tasmanian variety, of opium poppy was their target plant, and Tasmanian variety from the thibane it produces oripapine. So, what they have done, they block the coordinone reductase activity. So, accordingly they made the construct in this way. So, they use the RNA interference approach. So, the construct was made in this way. For example, if I write in this way, A, B, C, D, E, F, G, H, then again E, D, C, B, A. Now, so FGH so now what is happened? So, this is a sequence of the gene A B C D E, and then you put it in other direction A B C D E, in between you put one intervening sequence FGH. Then what will happen if this gene undergoes transcription? It makes something like this A, B, C, D, E, then E, D, C, B, A, and here lies your FGH. So, there is some problem with the board ink. I think now it is clear. So, it bends and form a hairpin loop. And so that means the mRNA and this is the gene. So, this forms the hairpin loop, and then eventually this portion will be chopped out and what we see is double stranded RNA. So, this double stranded RNA will be the target of an enzyme called dicer or dicer like protein, which will cleaves and make it small fragment, and then small double stranded fragment, and then eventually one of these will be removed, one of the strand will be removed. So, eventually it leads to the formation of short single stranded RNA or called short interfering RNA, and this short interfering RNA will be in as a part of this RNA induced silencing complex. So, where if an mRNA comes and then what will hold. So, when an mRNA for that particular gene will be produced. So, then what will happen when it is comes into the vicinity of this short interfering RNA, it will find the sequence similarity and sequence homology. So, that will bind. For example, okay, I will show only this portion. So, like this will come and bind. So, here this will come and bind, and as a result of that what is going to happen is that. So, let me delete this portion away, and as a result of that. So, this will be chopped away by the enzyme called Argonaut, and then eventually the whole mRNA will be destroyed. Now, this is in brief the principle of RNA interference. Therefore, the codinone reductase, in the RNAi construct of codinone reductase contains the coding region of codinone reductase, and then one should have a middle sequence, and then you again have the codinone reductase, and then this will be there under the control of promoter, terminator and all these things. And so eventually, when this will make the mRNA, so that will make the hairpin loop, and then eventually this whole mRNA will get destroyed. So, when the mRNA will get destroyed, that means the protein will not be produced. So, codinone reductase will not function. Keeping this basic, I just now go to the what has happened as a result of that. So, we will just go to the pathway diagram and explain. So, we start with say we start with reticuline. From reticuline, multiple steps it makes salutary denol, then salutary denol 7O acetate, and then it makes thibane, then oripavin, then morphinone, and then morphine. So, initially they use the Tasmanian variety, and later they also check this using the unique variety. So, that is why I put codinone CO. Codinone, which makes codine, and now I put the arrow. Okay. Laudanine, then you put laudanosine, and then codamine. So, razor dot pen laudanine to laudanosine, laudanosine to codamine, laudanine can also mix to this, and then sorry, this is scoularine, and the reticuline, this is called 7-OMT, which converts reticuline to laudanine, and then acid is here, acetyl transferase, and then here is the COR, coordinone reductase, COR coordinone reductase. I think I draw this step is, let me draw this better way, so that it will be clear. CO coordinone Okay. So, the coordinone reductase is here, now what we see, what has happened as a result of this suppression. So, as a result of this suppression, let us see what has happened in brief. So, in what we see, that the transformed plant, that is the RNAi, when RNAi mediated, when the gene was transferred, when this transgenic plants having the RNAi construct inserted and the control plant, that is the wild type. So, what we are going to see? We are going to see here the status of the alkaloids. So, the status of the alkaloids. Which alkaloids? One is the control plant, another is the transgenic plant having the RNAi COR gene inserted. So, name of the alkaloids we must write first. So, for example, what is the status of morphine? What will be the status of codeine? What will be the status of reticuline? What will be the status of codamine? We will also see the status of thebaine and laudanine and laudanosine. Okay. So, morphine in control, it should be very high, codeine should be present, control, reticuline will be very low, oripavin, you also write oripavin, oripavin will also present in the Tasmanian variety, and no codamine will be detected in the control or the wild type plant, and thebaine will be present, but in minor amount, and laudanine and laudanosine will not be able to detect. Now, what happened in the transgenic? In the transgenic, morphine very low or nil, codeine nil or absent, oripavin absent. Then reticuline will be very high. Okay, and then rather we will write high, not very high, we will write high. Okay, next is thebane. Thebane will be absent, and laudanine will be present. Laudanosine will also be present, and codamine will be codamine will be present as well. So, that means totally morphine, codeine both are absent, along with that thebane is also absent. So, that means this entire pathway is basically not operating. Instead, what is happening? It is producing high amount of reticuline, it is producing laudanine and codamine. Now, so that means by blocking codenone reductase, the impact we see on the upstream level. So, that is what is happening. So, when codenone reductase is blocked, that means is it something regulation called feedback regulation which is operating? Yes, in fact, the scientists they argued that this is one of the cases of feedback regulation which is happening with the specialized metabolism. And now, the T0 plant subsequently will be they had selfed and got T1, T2, and they found that this is stable. So, that even the T2 plant they did not produce morphine codeine, instead not even thebane, instead they are producing reticuline and the products derived from reticuline. So, this is one way of producing the licit alkaloid. So, that means there will be no illicit, there is no possibility of transforming them into drugs. Rather, there are different uses like codamine, laudanine, laudanosine, there are multiple uses for these products. So, this is a clear case of feedback mechanism. So, accumulation of reticulin suggests that. And so, what happens that after this gene silencing by RNAi, the precursor alkaloid reticulin, which is 7 enzymatic step upstream of codeine, got accumulated in the transgenic plants at the expense of morphine, codeine, oripavin and thebane. So, at the expense of morphine, codeine, oripavin and thebane, the 7 step upstream precursor reticulin got accumulated, and that leads to subsequent accumulation of the other products. So, this means that this worked very well. Now, what they also checked? They also checked as a result of this suppression that when the codenone reductase is suppressed, what happens to the endogenous gene of the pathway. So, that I am going to show you. So, before that what I am going to show you is that in the these are the standards. So, standards of thebane, laudanosine, laudanine, all these things and morphine, and this is the control where you see that maximum accumulation of morphine, codeine, all these things. Okay. And you see particularly in a few transgenic lines here. So, there is a accumulation of LS stands for laudanosine, there is accumulation of laudanine, there is accumulation of what is okay, and these products are forming okay, codamine and but there is no formation of morphine at all. So, this is the thin layer chromatography showing the product. Another interesting point I must say that the alkaloid, which is basically the precursor alkaloid, which is reticuline. So, what is the status of this reticuline? Is it S or R? So, what they have done is basically they ran HPLC from the transgenic plant, and then they took the they collect the latex and they methylated this whatever reticuline they collected from the transgenic tissue, they methylated to laudanosine. So, in other words, laudanosine is the methylated product of reticuline, okay. And then they use the chiral column and the both R as well as S standards of laudanosine are available, whereas reticuline standard is not that available. So, what they have done, they converted reticuline to laudanosine methylated in under in vitro condition, and then this laudanosine they checked whether it is R or S. So, what they found that this is basically S, no R. So, that means the reticuline formed here in the transformed plant is basically the S reticuline. So, that they have confirmed by converting the reticuline, isolated the reticuline from transgenic plant, converted in vitro to laudanosine, and then they ran the standard for both S and R laudanosine, and then they compare with the converted laudanosine, and they found that that is basically matching with the S. So, from there they confirmed that it is indeed S, and this is basically the HPLC chromatogram, which is showing that in the control you see that there is a lot of morphine, and whereas in the transgenic you see laudanosine, laudanine, codamine, reticuline, and in the transgenic so this is the this is the wild type the control, and this is the RNAi transgenic. So, here a reticuline accumulated, but if you see here in the wild type plant, reticuline is negligible. Instead these products are there, morphine in very amount. So, here morphine is almost nil or negligible, but reticuline enhanced you can see laudanosine, laudanine, codamine. Okay. Okay, so this should be codine. I think there is a mistake here. This should be codine. Okay, this is not codamine, but this is codine. This is their mistake at that point. Okay, and this is basically the pathway which I have discussed. So, there is no point. So, these alkaloids are called morphine and alkaloids starting from thibane onwards. So, what we see basically these alkaloids accumulating at the expenses of this route. Okay. And what they have also done, so this is basically the 7 and 4 are basically the showing the plant the DNA from the wild type. So, there is no NPT2 there, and here in this plant you see that coridone reductase is active in the 7 and in the 4 plant, that means the transcript is there. So, that means in the wild type plant coridone reductase is functioning, whereas in the RNAi transform plants almost it is negligible. So, this is very strong, and this is what is showing here. The blank means that here is the NPT2 gene is not there because of the wild type, but interesting point with is this, that you see all the genes remain unaffected, like 6-OMT, these all other upstream genes remain unaffected. So, even the berberine bridge enzymes, maybe it has more in these lines, but otherwise this is interesting. So, the endogenous pathway genes remain unaffected, particularly the upstream genes, but only the downstream genes. And when this paper was published, actually there was the demethylase genes were not available, that why they could not check it, like COM, CODM, T16OMT, all these things, T6OMT. So, those things are not available. So, that is why they have checked this. So, with this more or less I can end the class. So, basically this is a very successful case study. So, unexpected accumulation of non-narcotic alkaloid using RNA interference approach. So, this has lot of promise for the industry. So, with this I end this class. Thank you.